Hello students, welcome to this lesson. In this lesson, we are going to study transgenic plant production. Transgenic plant production is the process of introducing foreign genes into plant genomes to give them new traits or capabilities. This is a technique widely used in agriculture, research, and biotechnology. This technique or process has been used to make plants pest resistant, disease resistant, herbicide tolerance, drought resistant, to make plants improve their nutritional contents, to let plants to produce medicinal compounds, or to make them produce their own pharmaceuticals, to let them absorb heavy metals or pollutants from the soil or the environment, means letting them as a bioremediation agent. How can we genetically modify plants? To genetically modify plants, the first thing we must do is gene selection and isolation. The desired gene, which is responsible for a specific trait like pest resistance or drought tolerance, is identified and isolated from the donor organism. The second mechanism is vector construction. In this case, we need to isolate the vector from bacterium called Agrobacterium tumefaciens. The plasmid vector must be cut open by using restricted endonuclease enzyme, which was applied to cut off gene of interest. Then the isolated gene or gene of interest here is inserted into the plasmid DNA vector. Then the plasmid vector that contain DNA of interest should be cultured with Agrobacterium tumefaciens. In this case, Agrobacterium tumefaciens absorb this recombinant plasmid. Then this genetically modified Agrobacterium tumefaciens must be cultured with plant explants or plant tissue. In this case, Agrobacterium tumefaciens incorporate this gene of interest into the plant's genome. So transgenic plants regenerate in selection media. After certain time later, we can acquire transgenic plantlets with inserted gene.